So in this lecture, and this is the last one that I'm going to make for this section, the last PowerPoint presentation kind of speaks for itself. We will be looking at the other two types of muscle that are found in the body, smooth muscle and cardiac muscle. Smooth muscle is found primarily in it's found entirely in the internal organs of the body, with the exception being the heart. So it's the major part of most, or the major component of most of the blood vessels, the digestive, the respiratory, urinary, and reproductive tracts. Its function there is to contract and exert pressure on the contents of whatever tract or tube that it surrounds, and in that way help to move whatever the material is that's inside that tube forward. So some characteristics of smooth muscle: they are small in diameter, and they have kind of a spindle shape. I'll trace out one. Let's see, here's like the spindle shape. There's another one. So they have this spindle shape, a spindle, as you call it. The shape is sort of like this. They have a centrally located nucleus, and there's only one of them, unlike what we see in skeletal muscle. They are not striated. That means that the myofibrils are not arranged into sarcomeres. They're thinner, and they are more randomly distributed throughout the cell. And smooth muscle is always under involuntary control, and when it contracts, it produces slow, long-lasting contractions. So they're slow and they're sustained when we see smooth muscle contracting. So smooth muscle can be classified into one of two groups. There's what is called multi-unit smooth muscle, where the smooth muscle, each fiber operates independently of the other fibers. So this image here is showing multi-unit smooth muscle. There are few, if any, gap junctions found among the cells, so they don't pass the signal or the stimulus to contract amongst themselves. And we find multi-unit smooth muscle in the iris of the eye, in the erector pili muscle of the skin, and in blood vessel walls. So with multi-unit smooth muscle, individual neurons connect with perhaps multiples, but at least one, each one has its own source or of signal to contract, rather than what we're going to see in visceral or single unit smooth muscle. Visceral smooth muscle, again also called single unit smooth muscle, that's this kind here. They're found as sheets of densely packed, usually parallel muscle fibers, and they're held closely together by gap junctions. So we see the gap junctions in visceral smooth muscle. And so because they can spread that signal to contract throughout or across this sheet of muscle cells, they don't need to be each one innervated by its own branch of an axon. Frequently, they will display rhythmicity. That means that they can generate spontaneous on their own without, these spontaneous and rhythmically repeated contractions. And so this allows them the capability of something like peristalsis, where wave-like contractions will push the contents through tubular organs. Peristalsis, it's how we move food through the digestive tract, for instance, and how we move urine out of the bladder, urinary bladder. You'll get a contraction here, and then that is followed by a contraction here as this part returns to being relaxed again. And so the contents that were here get squeezed downward and downward by this wave of contractions followed by relaxation that will continually push the food ahead of that wave of contractions. So we find visceral smooth muscle in the walls of many of the hollow organs in the digestive, urinary, and reproductive tracts especially. Smooth muscle contractions are accomplished in a slightly different way, or a significantly different way, but the biggest difference is that rather than having troponin, they have a protein called calmodulin. The calmodulin binds the calcium as it is released into the interior of the smooth muscle cell, and that activates enzymes that then allow a muscle contraction to occur. So we see a really different pathway for muscle contraction to occur. Calmodulin binds the calcium, and when it does, it activates enzymes that then will cause muscle contractions to occur. And we get, as I said before, slower contractions and slower relaxation, but they can produce much longer and more forceful contractions with the same amount of ATP that a skeletal muscle would use to produce a shorter, less forceful contraction. All right, so that's the smooth muscle. Now, cardiac muscle. Cardiac muscle is found only in the heart, and some characteristics, they branch. So my kind of typical drawing for this is that you have smaller cells, certainly, than you do with skeletal muscle, but they branch, and those branches then interconnect, and so it's this interconnection that gives so much rapid passing of the signal to contract. The connections here are made with intercalated discs. Here is a image of, this is a scanning electron micrograph of an intercalated disc, and if you follow, you can see that the connection between the two cells is is really frilly, and I bet you can guess what that's about. Lots of surface area to allow lots of gap junctions so that we can spread the signal to contract from cell A to cell B very rapidly. Lots of mitochondria in the cell, lots of great blood supply, and always under involuntary control. They contract at a very steady rate, and they are incapable of producing sustained tetanic contractions, which is good. We want them to have to relax, because it's during relaxation that the heart can refill with blood so that the next contraction can squeeze the blood on out. If they were capable of tetanic contractions, then the heart could seize up and no blood could be allowed in, and that would be that. The person would pass on. Let's see what else to say. And that's it for this slideshow. So study well. If you have any questions, you know how to reach me.